Great. So welcome, everyone. Welcome to our next Berlin Digital Science for Drug Discovery meeting. I will give a few minutes of introduction and afterwards hand over to Andrea to share the first talk. So what are we trying to achieve with those events? So we want to bring everyone who's interested in digital science for drug discovery together across disciplines, also personally in Berlin. In the beginning, we had those meetings only online, but then after COVID, we were very happy to have them at the Free University again, where also the in-person meeting today is taking place. But we also kept the international meetings, so the online meetings, so that everyone all over the world can participate as well. So we will have scientific talks, 2 presentations, then there will be socializing, online actually as well as in person. So please do stay on after the last talk. So we'll put people into breakout rooms so you can meet some people, make new friends. So this series is organized by Clara Christ from Bayer, Andrea Vollkramer from Saarland University, Gerd Wolber from FU Berlin, as well as myself from the University of Cambridge and Pangea Bio. And so for next year, we still need to set the dates. So please come back to this website, digidrug.net, in order to check out the next few dates. And also let us know if you would like to present. So we will have those events every 3 months in Berlin as well as hybrid. So if you would like to present, please do let us know. So very happy to include you in the program as well. So the main meeting will be recorded. So I started that in fact already. And we will make the presentations available on YouTube afterwards. We agreed with the speakers, so we'll check that. And if you would like to access those recordings, please go to the same website, digidrug.net, and go to the past events link in order to go through the recordings of previous sessions. If you have questions in person, feel free to ask personally after the talk. So there will be the organizers and speakers who will coordinate that. And please repeat the questions locally as well, so people online can hear the questions as well. And online, if you have questions, feel free to type your question in the chat window. If you wish, also along the presentation already. And then afterwards, we will go through those questions to the extent the time allows. And after the last talk, just to remind everyone, please stay online, join us to the pub in person, or stay online to meet people as well. So we will have 2 presentations today, and those presentations will be by Francesca Grisoni on machine learning for drug discovery low data, and afterwards by Alberto Gobbi, who was with Genentech until recently, who now moved to Berlin, and who's very happy to present in the session today as well. And I will hand over to Andrea Vollkramer from Saarland University to introduce the first speaker. Andrea.
Thanks, Andreas. So I'm happy to introduce Francesca Grisoni. She actually did her PhD in 2016 at the University of Milano Bicocca. I hope I pronounced this more or less correct, on machine learning for ecotoxicology. And then she worked as a data scientist and, let's say, biostatistician for a pharmaceutical industry, before then doing a postdoc first at the same university in Milano and then at the ETH Zurich, focusing also on machine learning for drug discovery and de novo design. And since 2021, she is now an assistant professor at the Eindhoven University of Technology, where the work of her group focuses on, let's say, chemistry-centered deep learning methods. And we are looking forward to your talk on how you solve the data scarcity problem.
Well, solving is maybe a big word, but yeah, thank you, Andrea, for the introduction, and thanks to everyone for inviting me. It's a pity I cannot be there in person. Maybe next time I will try to be there in the audience. Okay, so let me share my screen. Okay. Do you see my presentation?
Yes.
Okay, cool. Thanks. So yeah, today I thought about using this occasion to talk about what do we do with low data in drug discovery, and use this also to introduce some of the work that's going on in my group and think about my professional or personal view about what we can do as a community to try and overcome the challenges of low data. Spoiler alert, I will mostly focus on the struggles of deep learning, or on let's say the deep learning angle. And I don't think that for this audience, we really need to speak about what deep learning is, but in case there's someone who's not very familiar with the term, the idea is just that we rely on these artificial neural networks that are a bunch of these neurons that mimic somehow how human neurons work. They take some inputs, aggregate and process it to provide an output. And when we stack many neurons on top of each other and next to each other, what we hope to do is to obtain models that can model very complex and nonlinear relationships between our inputs and our outputs. And as you know, they're getting quite some popularity in several tasks, and they have led to several breakthroughs, such as leading to algorithms that can play very complex games better than humans, or that can drive cars around without a pilot. And these tasks are very similar to what we want to achieve in drug discovery, right? We want to develop algorithms that will tell us, for example, what's the next move I should make? So what molecule should I make next and test next? Or where should I go in my chemical universe of all of the possible molecules one could make? But actually, we are dealing with a much more challenging game than, for example, chess or Go, because here in this case, we don't know the rules of the game, and we don't know, the regulations of our road, unlike the case of self-driving cars. But there's actually even more than this, than the fact of not knowing the rules of the game. And the problem, one of the main problems, as my title suggests, is that we're dealing with very low data regimes. So if you look at the breakthroughs of deep learning, in most cases, in those fields, there were a lot of data available for training, and we know that we often don't have that when we work in drug discovery. But also, very often we have incomplete information about the system. So for example, if we even just think about a ligand binding to a protein, very often we don't even know what's the pose in which the ligand binds to this protein. And it's also a very, very dynamical system. So there are many unknowns in our data, in the phenomena we want to study. And finally, even when we do have some information about our system, then we somehow have to convert it into machine-readable formats. And very often we have to lose some of the information about our system. And this is very different compared, for example, to the field of image processing, where what we have to deal with is an image. So an image can be read by a computer. In this case, we're dealing with real entities that we can only represent in somehow an approximate way for computers to learn from. So what I really want to focus on is where I think we are when it comes to deep learning compared to machine learning approaches, and whether there are some strategies we can put in place to overcome partially these limitations. And so, as you probably know, one of the simplest way of applying these machine learning and deep learning models in drug discovery is to use a so-called quantitative structure-activity relationship approach. Let me pick a pointer. And basically, the idea is that we assume that everything we need to know about a molecule is contained in its structure. And if I have a sufficient number of annotations, for example, on bioactivity, I can train machine learning models to try and connect these two sides of the equation. So to predict, given a structure, how active it would be or whether it is active at all. So, and before the, let's say, advent of deep learning with the traditional QSAR, the way in which it was achieved and still achieved is to convert somehow this chemical information into numbers that are nothing, and we call them molecular descriptors. And these numbers are nothing but human engineered features that are obtained based on what we know about the system. Well, deep learning allows us to partially bypass the need of these descriptors and allows us to deal with more, let's say, raw representations of the underlying chemistry, such as, for example, molecular graphs, where each atom is a node, and these nodes are connected by edges, which would be the nodes, the bonds, and then we can also specify some properties for these graphs, or even SMILE strings that are basically string notations that capture which atom is present and how they're connected to each other. And what we see is that in many domains where deep learning has led to breakthroughs, actually bypassing the need for human engineered features, what is one of the elements that really made a difference in the performance of deep learning algorithms. So one of the main questions we wanted to answer to when I joined Eindhoven was whether this is really the case when it comes to molecules and drug discovery. And so to achieve this, we really wanted to push our analysis and see how far we can go with this method. So we decided to look into what people refer to as activity cliffs. And activity cliffs are nothing but pairs of molecules which are quite similar in the structures, but where a small change actually leads to quite a big change in the experimental properties, such as the bioactivity of these molecules. And usually in the field, what people say is, okay, whenever you find activity cliffs, just run away as far as you can and escape from these activity cliffs regions. And I do agree partially with this, because in fact, we know that the way in which a ligand and a protein interact is very complex, and we have a bunch of complex interactions. And sometimes even by changing a tiny portion of the molecule, such as we change something and we lose a hydrogen bond, might lead to very drastic changes in bioactivity. But it's also true, on the other hand, that if we had a perfect oracle, a model that would know exactly how each of these elements changes the bioactivity, then activity cliffs would not actually be a challenge. It would be just one of the possible cases we might have present in our data set. So we really decided to use activity cliffs as a way to test our models and compare how the different approaches would work in these cases. So Derek, a very talented PhD student in my team, started to then collect and curate data sets from Campbell, define and find activity cliffs, and then compare different machine learning models on these cases. I will not go too much into the details, but he tried several types of, let's say, traditional machine learning approaches based on descriptors, with 4 different sets of molecular descriptors that would capture different aspects of the chemical structure, and also several deep learning based approaches. 4 based on graphs. I don't think it's important to discuss these labels. Basically, they just differ from each other based on how they assemble and aggregate information on these molecular graphs to make a prediction. And then also approaches that learn from the string to the SMILE strings to perform a prediction on bioactivity. So you can find the full deck of results in the paper I cite down there, but I decided to focus mostly on deep learning. So what we saw all across our benchmark is that indeed activity cliffs are a very, very challenging case for all of the approaches across the board. But if we focus particularly on the comparison between deep learning methods, which you see here in green, and let's say traditional machine learning methods, here I report just ECFPs. These ECFPs capture the presence of substructures. We see that actually this is an error, so the lower the better. Actually, deep learning models tend to struggle even more than traditional models, traditional approaches with these activity cliffs, which is a bit counterintuitive if we think that they have access to the full structure and not to human engineered descriptors. So there's still something to do there. But also what we found, I believe, was even more interesting is this. So when we looked at the predictions on different targets, and compared, for example, the error on all of the molecules we had in our test set compared to only the error on only the activity cliffs, we saw that in some cases, we would have something like this. Didn't matter the difference between the errors, but we would have somehow a linear relationship between the overall error and the error on activity cliffs, which means that actually, if we pick a model that performs overall well on our test set, we would also have good chances of picking a model that performs proportionally well also on activity cliffs. But there were also cases like this one where the situation was really a chaos. And the risk here is that we might end up picking a model that seems good on the overall set of molecules, but when you look at its performance on activity cliffs, it actually is very, very bad. So it might perform very bad prospectively. So we started to ask ourselves what does differentiate between these cases and these cases? And maybe some of you already have the answer, but what we found out is that it boils down in many cases to how many molecules we have available for training. And basically we saw, here we're looking at correlation. So models, it doesn't matter what the difference between the errors is, but like how much we can imagine the errors on activity cliffs based on the errors on all molecules. And we see that when we go to low data regimes, here is truly, truly a mess. So in some cases, we might still be lucky. And in some cases, we might end up picking models that perform very badly on these activity cliffs. So in the end, one of the take-home messages is that activity cliffs are not really enemies, but actually they are something that can help us understand how these models perform. But going back to the low data talk, I think this one, again, shows how much we are actually struggling whenever we deal with low data, and sometimes we might not even realize it. So one of the things we're trying to do now is to ask ourselves what strategies we can put in place to be a bit better when it comes to very, very low data regimes. And one of the ongoing projects is about active deep learning. And actually, active learning is not a new term in the drug discovery domain. And the idea is that we try to change a bit the way in which we use this model for prospective applications. So normally, one would have a training set. You use a training set to produce a model, and then you use a model to screen a large library, for example, of molecules one could purchase and then test in the wet lab. But with active learning, what one can do is instead to use, of course, the model you have, the molecules you have to train a model, use the trained model to select some molecules, usually it's fewer than if you were using the traditional approach, bring these molecules to the wet lab, and then use the results to update your model. And the hope is that by doing this in a smart way, one might end up with a model that gets better and better and can guide you to your goal faster. That's the idea. And actually, as I said, active deep learning is active learning in general is not novel, but what we wanted to do here was to really try and push this model to the very, very low data regimes and see how, sorry, the light turns off on me here, see how these active learning approaches would perform when we start from very, very little data. So in this case, we decided to apply the models to very large data sets for which we had the labels. So it was a retrospective study. And we actually studied a few factors that might be important whenever we apply deep learning, active learning in practice. So here, as I said, we wanted to start with very low data regimes. So we started with 64 molecules that would be then used to screen these large libraries. And then we also wanted to understand what's the effect of the diversity I have in my starting set. If I have molecules that are very, very similar to each other, for example, a set of congeneric compounds, would I still be able to effectively explore this chemical space and find hits? Also, we wanted to investigate the effects of the models that we choose. And in there we have, again, ECFPs, so fingerprints, compared to graph neural networks based on that learn directly from graphs. And then, of course, there's also another question. Once we have a trained model, how do we select which molecules to bring to the lab, or in this case, to label with the actual labels that we have in the data sets? So we set up this pipeline with all of these factors, trying to understand mainly two things. First, does active learning work when we have these very, very low data scenarios? And also, what are the important factors that we should consider whenever we want to apply this in practice? And actually, there are a few nice things, or at least I find them nice, that we brought home from this study. And one of the first ones, so basically is what you see here when you compare left and right these two plots. And basically what you see here, just to ease you a bit into these plots, is the enrichment factor. So we draw from the library, and we basically check, wait, sorry, there's a bit of interference. Okay, gone. So basically we draw from the library and we check how much better we're doing in enriching it in bioactive molecules compared to just random picking. And this is what you see on this. So 3 means 3 times better than random. So if you compared ECFPs with graph neural networks, what you will see is, again, that even when graph neural networks are allowed to choose from which data they can learn next, they still perform worse than extended connectivity fingerprints. So again, I was hoping it would be better, but there's still something to do there. But one of the other things that I think is very interesting is that how we decide to pick the molecules, and these are the colors you see here, so the different acquisition functions we investigated, how we decide to pick the molecules is what really drives our performance. And there are a few of these acquisition functions that really emerge as a very good idea when picking what to learn from next. Also, an analysis that Derek did was to investigate how good these models are in picking up substructures that are relevant for the bioactivity of interest that were not included in the training set. And here you see 2 cases, actually. This one is a fragment that is relatively more abundant in the hits in bioactive molecules than it is in non-active molecules. And here a fragment that is just equally distributed among the 2 classes. And here again, we see that if you're very smart in how you pick what to learn next, what to learn from next, in some cases, the models can really pick it up and start emphasizing these substructures in the molecules they pick. While in some other cases, if you don't choose your molecules wisely, you might just start picking from things that are just there, but not necessarily relevant. So I think this is really nice. But probably the most unexpected take-home message that we got from this study is when we investigated the effects of the starting set. So as I said, we investigate different levels of diversity, and we want to see, okay, am I really going to fail if I have molecules that are just too similar to each other when I want to use the model prospectively and enrich my list in active molecules? And the answer is actually that this is not the case, and we were not expecting it. So what you see here is how similar the molecules that are picked across cycles are to the starting molecules. And what we see is that no matter the start, some starts are a bit less advantageous than others. In only a few cycles, the model can rapidly, let's say, balance the information they have, and they start picking from molecules that are relatively new compared to the starting data. So it doesn't matter how unlucky you are with your starting set. It might take a bit more, but overall, these models can compensate from the lack of structural information in there. So I think this is really nice. And with this few insights that we got, we're now bringing these models to the wet lab in 2 applications. So I really hope that we will have some interesting results from that side too. And just as an advertisement here, we also applied these ideas to nanoparticle formulation, where we had this automated pipeline of formulation, imaging, and then feeding the information back to the active learning pipeline. And even here, we saw that active learning is really helpful, and it allowed us to rapidly formulate interesting nanoparticles. But I will not go too much into the details because of the story here. Yeah, and as I said, I hope we will have very soon some interesting results about applying this in a prospective scenario. So yeah, if we reflect again on this, let's say QSAR, where basically we connect the structure with the bioactivity in some sort of forward manner, my opinion is that we're not there yet when it comes to deep learning and how much better it performs compared to, let's say, more standard and traditional approaches. But I do believe that it is a very different story when we go the other way around. So when we perform de novo design. And the idea of de novo design is that instead of mapping a structure to the bioactivity, we actually start from bioactivity we're interested in, and we ask the model to generate structures that could hopefully match the bioactivity we intend to achieve, for example, be active on a given target. And there are many approaches now, so it's starting to be an old field now when it comes to deep learning for de novo design. But in my opinion, one of the most successful ways of tackling this problem is being inspired by natural language processing. And basically, now that there's ChatGPT, there's no need to speak too much about natural language processing. But the idea is that if we show models with large corpora of elements of language, of examples, then these models can pick up elements of this language and can learn, for example, the probability of occurrence of a word in a sentence or a sentence in a language, and they can also be used to generate new sentences. And when we think about chemistry, we can think that such a thing as a chemical language exists. So, for example, if I gave you this molecule and asked you to move the atoms around randomly and try to connect them with bonds, we would most likely end up with a molecule that does not make any chemical sense. So there is such a thing as a grammar or a syntax to this language. So how do we need to put the atoms there and connect them to obtain a molecule that can be actually made and make sense? But also what we're interested in when we work in drug discovery is actually the semantics of our language. So what do we need there for this molecule to have the properties we desire? And actually, the way in which we can translate approaches from the natural language to approaches for chemical language modeling has been there for decades. And this is constituted by string representations, such as my strings. And as I said already earlier, the idea is just that we have a molecular graph and we somehow traverse it in any direction we want and just annotate what atoms we have in there. And then we give special characters for branching and bonds and so on and so forth. So we can convert chemical information into a string. And the idea is that in the same way in which we use these models for language, we can train neural networks of various sorts to basically learn from smiles and predict at each step of this sequence what characters should follow. So basically, once we train them, they will also be able to generate new smile strings, one character or one element at a time. But you might already be thinking, like asking yourself, how do we still make it work in practice when we don't have a lot of data? And there are many approaches out there, but one of the things that has worked quite well in the past is to use transfer learning. And the idea of transfer learning is that if you don't have enough data to train a model from scratch, for example, imagine you want to use bioactive molecules, but you only have dozens of them, you can rely on some sort of other data set that is bigger, but still relates to the task you want to perform. So already in 2018, when I was a postdoc in Gisbert Schneider's group, we used this transfer learning. And the way in which we did it was that we first pre-trained our model with smiles that were obtained from Campbell. And it was 300,000 smiles of bioactive molecules. And we would just show them to the network and train the network to predict what characters should follow at each position. And what was interesting was that already after this very generic pre-training step, if we ask the model to generate new smile strings in this way, one element at a time, it would produce more than 90% of smiles that were chemically valid. So it means that the model could learn the syntax or the grammar of this chemical language. But then, of course, what people are usually interested in is to focus this model on a smaller set. And so basically the idea is that once you pre-train, you just take the data out, put new data in, usually on bioactivity, and then you keep training your model. And this is one of the tricks in which you can basically compensate a bit for the lack of data. And I will show you that it works in a second. And another trick that is very handy when it comes to smiles is the so-called smiles augmentation. And even this has shown to work quite well. And the idea is that, as I told you before, you start from any atom in the molecule and you traverse the graph in any direction you want until you have touched all of the nodes of this graph, which means that actually every molecule could be annotated with many different smiles. And so, for example, if you have 10 molecules, you could obtain 10 smiles per each molecule. So then you would move to a data set of 100 molecules instead. And this has shown to actually improve the quality of the information that these models can learn. I will not really go too much into the details, but actually we have applied these concepts together when I was at ETH to bring these models to the wet lab in many different applications. Here I list only some of them. And if you see, we are already in quite a low data regime. So we're using a few dozens of molecules for fine-tuning. But I decided to speak about one of the most recent studies that was led by the group of Daniel Merck. And I did, spoiler alert, I did very little for this study, but I think it still fits very nicely. And basically what they wanted to do in his group was that they found this agonist, which was the first, let's say, annotation for a potent agonist that they had on the target. And they were interested in understanding whether these chemical language modeling approaches would work even with just one example. And basically, the first attempt was about using this standard pipeline where one can just pre-train with Campbell, with bioactive molecules from Campbell, and then fine-tune using this agonist. And I don't know if you have a pre-formed idea in your head, but this didn't work very well. And one of the struggles was really to make the model get out of the, let's say, scaffold that was the scaffold of this molecule. But the trick that ended up working was to fine-tune again with some weaker agonists that were available in his group. And this basically showed to help the model get out of its own narrow knowledge on this potent agonist and still allow to create some diversity. And when these molecules were brought to the lab, they were able to obtain two new agonists to expand their set. And I think this is a very, very nice application of these models in the very, very low data regime. But of course, some of the problems are still there. So for example, how do we push this diversity further when we have only a few examples? So yeah, my personal or professional opinion on this is that we, let's say, when we focus especially on deep learning, we're doing decently well when we have enough data to learn from, and we're getting better and better in generating new molecular hypotheses when it comes to de novo design. So I think it's a very, very nice and vibrant moment to be working in this domain. But I also think that we have just started to scratch the surface of what we can do. So we still lack tools to be able to actually really control the de novo design process. For example, if you want to generate structurally novel compounds, and also whenever we go to the low data regimes, it is very, very tricky to predict bioactivity. And of course, one of the holy grails or very cool research topics to work in would be, how do we do it when we have just no known data for a given target? So with my team, we're trying to approach some of these questions, and I think that these are two of the main things that we can do as a community. One of the options is trying to learn better information from what we have, from our current way of representing the problem, or we can try to incorporate more information so that these networks have more accessible for them to learn from. And for example, when it comes to learning better information from what we have, there is this preprint that we put out a few weeks ago, led by Reza, where we reflected on how these smile strings work. And very often you find that elements that are close in the graph end up being very far in the smile string. And this information is basically lost when we use networks that just keep track of one character at a time and generate one character at a time. So we borrowed these S4 models from the computer engineering domain, which can deal with long-range dependencies and see all of them together and still generate one atom at a time, one element at a time. And they seem to be very, very promising for de novo design. And now we are expanding them to see how far we can push them. But then actually what I think is even more interesting is number two, the fact that we do have a lot of knowledge on the chemistry. For example, if you look at these smile strings, but actually we present this knowledge just in the form of strings. So it's a very basic way of representing a very complex phenomenon. So what we're working towards is how can we infuse this representation learning process with something more about the chemistry of the process. And we're going to having representations that are closer to the chemical reality or what we know about it, up to trying to infuse biophysics and molecular dynamics information in there. So hang in there, stay tuned, and I hope I will have some interesting results on this soon too. So with this, I am done. I hope you enjoyed the talk. I have many people to acknowledge, my team and also our collaborator in Munich, all the funding agencies, and mostly the DigiDrug organizers for inviting me. And I'm happy to take any questions you might have. Thank you.
Thank you very much, Francesca, for a very interesting and very opinionated view also, which I really enjoyed. I think we will start with a few questions from the chat. And I will just read them out to make it a bit easier. David Marshals asked, how low could you go with low data? So how few molecules could you start with?
Yeah, so with the, if it's about the active deep learning part, I think we have tried even with 32 molecules and it didn't make a big difference. So we decided to go for 64. I think that a few dozens would be okay. And in the end, what we see when we look at the similarity plot is that these models compensate very quickly for lack of diversity. So I think you just need some examples to try and steer the model in the right direction. So it would be interesting to try with, I don't know, 2 molecules, one active and one inactive, and see whether the models manage to get out of that. I don't have an answer now on this, but yeah.
Thank you. Maybe I give the word to Berlin, because Clara just said that there are some questions in the room.
Thanks, Andrea. So we have at least one question. Oh, okay. Hi, this is Giulio Napolitano from Berlin. So I have a sort of a comment and a question. So one comment is I would be very interested in seeing how the experiments with large language models will go, because first thought I would think, you know, that's a bit of a long shot, because I think that the very power of the LLMs are because they are using language, natural language, and they are actually sort of leveraging the real meaning of words, which is the context, you know, how they are in the context, how they're used. And I don't know how this is going to be transferred to something which is maybe analogous to a language, but still it's not a natural language after all. I would say maybe this is a bit better in omics domain, but I'd be curious to see, you know, how it goes, to be honest. I don't know if you have any thoughts on this. And the other question is, sorry, the question actually is, so embedding knowledge, as I see one of the approaches you have is representational learning, but I am a big fan of reinforcement learning. Have you thought about how you can embed policies for reinforcement learning in this
Yeah,
framework?
so we have a few projects on reinforcement learning too. And I do think that reinforcement learning is quite powerful in allowing you to steer the model where you hope it might end up, you know, like where you want it to go. But the problem we have in drug discovery is that we do not have oracles that are trustworthy. So for example, if you just look at bioactivity, you could say, okay, I use reinforcement learning to steer my model in regions where the molecules are predicted to be bioactive. But actually your scoring function is biased. And there are a few studies that show that these models can easily, if not applied properly, can easily exploit shortcuts and the weaknesses of your oracles to actually cheat the system. So I do think that, for example, in the Nova design examples I showed, that this self-supervised and auto-regressive type of learning, where you don't explicitly force a function on top of it to steer the model, is actually a bit safer from these types of shortcuts than reinforcement learning. But yeah, in some cases this can work. Yeah, absolutely. I don't know if that answers your question.
Yeah, thanks.
Okay.
He's happy.
Okay.
Thank you.
And there's one more question from Alberto.
Yes, exactly. You want to come to the front? Then actually we have a chance to people see you. No, yeah.
Okay. Hi, Francesca. Thanks.
Hi.
Okay, here. Thanks for the great talk. I was wondering, when you presented the activity cliff work, you showed that there's a strong dependency on the number of molecules in your training set. Have you also checked if the dependency is maybe just limited to have at least one example of that specific cliff in your data set? Because that seems like something you could learn from pretty quickly.
Yeah, we did try to check for many factors, and when we split the data, we try to make sure that pairs are separated. And I don't think we explicitly check for, okay, is at least one example of this specific case included in the training set. But this is probably a very good control that we could perform to see if there's something more. Yeah. Thanks.
Thanks. Back to you, Andrea.
Thank you. I will use my power to also ask a question. You nicely showed this the potential of active learning or active deep learning. Now you're fully
yeah, the lights. Yeah, I'm too still apparently. Should gesture more, as all the Italians do. I forget sometimes. Sorry, go ahead.
Yeah, and you showed that you had different acquisition functions, and I think two of them worked better in your experiments, and I forgot which were the two. So in a prospective scenario, like, would you settle on one of them? And if so, which one? Like, would you go for uncertainty or for exploitation?
Yeah, so the two ones that worked better were, so one was based on just, and this was also something we were not expected, on exploitation. So picking things the model is sure about, and we're expecting that after a while you just saturate what the model knows. So we were expecting exploration to be better, but that wasn't the case. And the other one was using mutual information. So trying to pick information that was relatively new for the model to access. Yeah, so we will just pick one, and we will pick ECFPs again. And actually they perform equally, so we'll need to do a few analysis rounds to see if there's any function that seems to be better. But yeah, I would stick with one.
Thank you. And then there are more questions in the chat. I think we still have a few minutes. So the first one is from Nir Sharp. Do you have any comment about possible applications of machine learning or AI in the context of polypharmacology?
Oh, yeah, that's a good question. Well, yeah, I mean, this is also something we aim to work on. Either, well, it's the same, either polypharmacology or trying to modulate the selectivity from a computational point of view is the same. You just have an array of targets and you decide if you want to modulate all of them or only one of them. And I do think it is very promising, but for the current approaches we have, we're not there yet. Not at the point where we have really a fine-grained control on the properties. We see that, for example, in these de novo design projects, if you have dual inhibitors in your fine-tuning set, you might also end up with dual inhibitors in your designs, but not in the way we would like to say, okay, on, off, or every target on, for example.
Yeah. Thank you. And one more from Pier Michele Kobauri. What is your view on the combination of physics-based modeling and machine learning? Do you have experience with mentoring the machine with more scientific knowledge while it learns from data?
So I don't have a lot of experience when it comes to physics-based stuff. It's a bit out of my out of my expertise, but I do think it's really promising. And actually, I'm more oriented towards chemistry infused or chemistry inspired types of algorithms, but I think it's the same idea. So we have gathered so much knowledge from the system, physical, biological, chemical systems, that it's a bit of a pity to not infuse them into our networks and just use a graph or smiles, which are very limited representations.
Thank you. And then I think we have one more question in Berlin, and then.
Yes, thank you. You also want to come to the front, or I can also, no. By this.
Oops. Francesca, hi Francesca. Here's Manling. My question is, with the active learning, I mean, it's really kind of like amazing that it will be able to kind of like starting with 64. Have you tried actually with the peptide? Because that's actually kind of like the holy grail is where you have even less data. And so it would be actually already great if you can start with 64.
Mm-hmm.
I mean, small molecule, you can easily get up higher. So
yeah, so we have an ongoing project with some collaborators here at the TUE where we're developing models to predict the bioactivity of peptides, and then we will bring them to the lab. And our idea is already that if we're not successful in the first round, we will just plug it back to the model and keep going for a couple of iterations. So that might work actually. I don't know, but yeah, definitely it's very, very interesting as a topic. Thanks.
Thank you. And then the final question from the chat. There's one from Ayaz Malik. So before building models, do you calculate the descriptors of the molecules? And if so, what method did you use and what type of descriptors?
Yeah, so I think there are different levels. So in the standard pipeline, you have your structures, you create them. When you are sure as much as possible that they're correct, you can compute descriptors, so you obtain numbers. If the question is about the activity cliff paper, we couldn't test all of the possible descriptors, but we went for groups of descriptors that would capture substructures, like these ECFPs, groups of descriptors that would capture 3D properties of the molecules after you optimize their geometries, and then physicochemical descriptors and other descriptors that capture the 2D information to have a bit of a broad view on what we encode. I'm not sure this answers the questions, but yeah, that's That's the approach.
Okay, thank you very much. Thanks for the talk. Thanks for the Q&A. And with that, I would hand over to Clara, and she will take care of the in-person Berlin part of the meeting.
Yeah, welcome from our side. This is Berlin speaking. We have a nicely snowy night here in Berlin. And it's my pleasure to introduce Alberto, who recently moved to Berlin. And he has obviously a very good taste of cities, I must say. And he has an intro slide, so I will be very brief. Alberto will be talking to us about fast quality strain energy with neural net potentials. And yeah, with that, I stop here and I hand over to you.
How is this? Can you hear me?
Yes, thank you.
Okay, thanks. I can do it, I think. Okay, so thanks for having me. Thanks for inviting me. And the reason I want to give the introduction slide myself is because I'm not only introducing myself, but also Manling Li, who is here and happens to be my wife and happens to be working with me for the past 20 years or so. And we are both happy to be in Berlin. We moved here to be closer to family. And I'm excited to meet everybody. So this is a brief recap of our past. We've spent the last 15 years at Genentech in San Francisco. Before that, we were at the small startup Anades Pharmaceuticals in San Diego for 5 years, of which Manling spent 3 years in setting up her own company that developed open source software. And before that, we were in Basel, Manling at Roche, and me at Ciba, that became Novartis, and then Syngenta. And before that, we studied chemistry in Marburg. Manling did her PhD in NMR and me in quantum chemistry. Our background covers software development, including things like compound registration, biological and peptide registration, UI development, data browsing, and integration of computation tools in Mo and PyMo.
Can I interrupt quickly?
Yes, I'm hearing there is some sharing problems.
Yeah, the slides are not moving in the shared version. No, we also don't see the presentation mode, just the other one, and it's, yeah, not moving. Yeah, perfect.
But now we don't see you, but we really would like that. Yes. Okay. Can you shift the window a bit to the right? Maybe divider. Okay,
Yeah, we don't. Yeah, that's good. Thank you. Okay, I was just giving a little bit of background. And then there are things like property calculation and more computational tools like the strain energy I'll be talking about today. Now I cannot move anymore. Okay. We've also done project work as computational chemists in MedChem projects, data analysis, eating some of our own dog food with the cheminformatics tools that were developed. And specifically, Manling has done some software project management. With that, I want to acknowledge that the work I'm going to present to you about the strain energy work was part of an incubator project at Genentech, where I could spend 2 years of my time doing science specifically to that, but it was also a collaboration with lots of people at Genentech and also outside of Genentech, as you can see on this slide. So now how do we define strain energy? If you think about a molecule binding to a protein, it'll make some positive interactions with that protein, but to do that, it might actually have to deform from its natural state. And if you think of the molecule like a spring, you can see that in the upper graph, you'll have to put quite some energy into deforming that spring so that it fits into the protein. While if you are able to preform the molecule to adopt a shape that is complementary to the protein, you are actually not going to have to invest that energy. And so your strength of the bond will be stronger and you'll have a more potent compound. And so it's an important component to the binding energy of the molecule. This is a little bit simplified, the potential energy surface on the left, where you have a binding hypothesis that is on a higher level of the protein, of the potential energy surface. And the difference of that energy of that binding hypothesis to the energy of the global minimum is what we call strain energy. If you plot this in 2D, you see what is on the right, where you have the distance to the input on the x-axis and the energy of the y-axis. But what I want to point out is that it's not just the total energy difference that is important to know. You also want to know what the slope is in the beginning of that curve, because if you have just maybe a bond length that is slightly off in your binding hypothesis, that's a big energy drop if you relax that bond length, while the change in geometry is very little. And so when you develop a method to compute the strain, you want to take that into account. And this is the workflow that does this, that we've been using. It follows work by Borstrom and others from 1998. Essentially, what you're doing in the upper part of the graph is you're doing a full-scale conformation analysis of your input and calculating the lowest energy, the global minimum. You're calculating a local minimum, which is just minimizing your input to the lowest energy that is close by. And then we're also minimizing with some potentials that counteract the relaxation. And these potentials, we vary the strength so that we see various conformations that are close to your input, but somewhat relaxed, so that you can actually see where the strain is in your molecule. And here's an example of starting from a PDB structure of a RORC inhibitor. The energy difference to the global minimum is 40 kcal. This is the input structure, so the distance from the input is 0 angstrom. And if we look at how the relaxation happens, when you apply this potentials that counteract the full relaxation, then you see that even just a relaxation of 0.15 angstroms drops the strain to 5 kcals per mole. And then if you relax a little bit more, you see there's a little bit more deviation. Now we have 0.4 angstroms from the input, and the strain energy is 1.5 kcals per mole. I want you to remember this 0.4 angstroms, because that's what we decided is the amount of relaxation that we will be allowing to define what the strain of a molecule is, because it's still very close to the input, but allows a significant amount of relaxation of less than relevant parts of the molecule. And if you look at then the further relaxation, you start seeing that there's a stronger deviation from the input, and the conformation cannot be considered quite identical to the input. And the global minimum in this case has still some similarities, but it's significantly different from the PD conformation in the PDB. You can do this now for lots of compounds from the PDB to get the statistics on how they behave. What you're seeing in this box plot here is 1,100 neutral compounds from the PDB that were allowed to relax 0.2 angstroms from their conformation in the PDB database. And you can see that about 50% have 2 kcals or less strain, and 75 have about 5 kcals or less strain, if you allow for 0.2 angstroms of relaxation. If you look at different amounts of relaxation, it's obvious that you'll have, if you allow more relaxation, you'll have a lower distribution of the strain energy. And if you look at the 0.4 that I am suggesting to use, you'll see that 50% have less than 1 kcal in strain, and 75% have less than 3 kcal in strain. So when we run these calculations, we tell our chemists, okay, if it's more than 3 kcal in strain, then you might want to really figure out where's the strain. Is this a real thing? Is this an artifact of the method? Can you reduce the strain to make your compound more likely to be a good compound? We've integrated this into Mo, so from a binding hypothesis with a single menu click, the strain energy can be started. And if you use a force field, this takes just about 20 seconds. And what you get back is essentially multiple conformations. You get the input with its energy relative to the global minimum. You get this intermediate constraint minima, the local minimum, and the global minimum. And by looking at the intermediate steps, you can figure out which part of the molecule contributes most to the strain, and which should you address if you want to design a compound with a smaller strain. Here's an example of a project where 2 enantiomers were crystallized. They differ in the stereochemistry on the lactam ring, where the amide group is pointing towards the top. In one example, the top is actually hydrophobic, so you wouldn't expect an amide in that region, while the bottom allows the amide to actually make some nice interactions with hydrogen bonds. However, if you look at the potency, the one that is actually facing the amide into the hydrophobic pocket is actually 2.5 times more potent than the other enantiomer. But the strain can explain at least part of this, because you can see that the lower examples has about 3 kcals in strain. And so that could be a contributing factor to the difference in potency. Now, let's switch to neural net potentials. I know that there are groups at the university in Berlin that work on neural net potentials too. This is a lot of fun. And what you're doing there is you're training a neural network to take atomic coordinates of a molecule and spit out an energy. And you train it on quantum mechanical energies. And if you choose a good quantum mechanical method, then you get very accurate energies. We've been working with Adrian Rotberg and his group on this. So we've used the ANI2X dataset that has 9 million conformations for about 60,000 compounds at the DFT level that is given on the slide. The results that you're seeing in this slide will be with an in-house developed method. By now, there is Torch ANI that you can download from GitHub, but you can also download all of the code from Genentech on following these links. Now, do this method work? The graph on the right here shows the correlation between the quantum mechanical absolute energy and the energy computed with such a neural net potential. And if I was seeing a graph like that, I would say I don't really believe it, but the details is actually in knowing what is graphed there. What you're seeing here is actually a range of about 1.5 million kcals per mole. So even a very small difference on here would make a very big difference for us. Now, if you look at relative energies, that's like zooming in on small difference, then you see that the correlation is not quite as good. And this feels actually more real to me, I must say. The correlation is not excellent, but it's still pretty good. You have an RMSE of about 1.5 kcal per mole. And this is definitely very useful. And obviously, the scale of performance of such a neural network compared to QM calculation is pretty amazing. If you want to calculate strain energies with that, you use the workflow that I showed you before, but you replace, in the workflow I showed you before, we were using OpenEye tools, ShiftKey to run MMFF94 minimization, and here we're using an in-house developed optimizer that uses a neural net potential to minimize the compounds, the conformations, and you get essentially the same workflow. This now takes about 5 minutes on a GPU, so it is slower than the force field. But if you wanted to do something like this with 1500 minimization with QM, you would spend a lot of electrons and a lot of time. Does it actually work? Does it give you better results or useful results? To evaluate that, what we did is this thought experiment. Let's assume you have a DFT potential energy surface, which you trust, and you found a global minimum on that DFT surface. Now, how do you compare other methods? And what we decided to do is we asked the question, how frequently do I find a minimum that is close to the global minimum on the new potential energy surface? And if you have two methods, you can compare it just by counting the frequency. Now, this criterion has two parameters that define the box size in which you are looking for your minimum. And that's the one side is the energy threshold, and the other side is the geometry deviation from the DFT minimum. And we've run the experiment with our in-house ANI force field neural net potential and with the MMF94. And if you look at with different box sizes, so what you have on these axes is the geometric threshold on the x-axis and the energy threshold on the z-axis. And if you look at the very small box size, those are the two bars on the right front left. You see that there's about a 50% higher frequency of finding a minimum in that small box with the neural net potential as compared to the force field. And if you go to intermediate box size, there's still a significant difference, while if you make your box infinite, the difference goes away. So there seems to be an advantage, at least in these cases. Here's a comparison straight of the strain energy calculation between the neural net potential on the left and the force field on the right. The distributions are very similar. What you see on the right is a graph that plots the strain with a relaxation of at most 0.4 angstroms. So that's what I was talking about before. And for the force field on the y-axis and the neural net potential on the x-axis, and you have the quadrant on the lower left and the upper right where the two methods kind of agree, and you have the boxes that are on the here and here where the two methods disagree, and those are the examples we can use to understand which method has which limitations. So let's look first of the examples where the these are PDB structures again, so they are assumed to be low energy. And the force fields for these three examples, the force field thinks they are strained as deposited in the PDB, while the neural net potential thinks that they are not strained. On the top right, the 5JN8 example, the difference between the force field, I should say, the PDB structure is in gray, the neural net potential in blue, and the force field in yellow. And the difference between the force field and the neural net potential agree, while the force field flips the emmet in this example. And this is an example that shows that many force fields have problem with sulfur-oxygen interactions that are assumed to be repulsive in force fields, but in reality, the sulfur is so polarizable that they are actually attracted. And so this is a problem, a known problem in force fields. In the example in the middle, we have a torsion around a phenyl ring that is conjugated to two emmet systems. And the difference here is that the force field thinks that there's a 6 kcal energy barrier in that rotation, while the neural net potential finds a single 0.1 kcal in barrier here. The most interesting one, I think, is the one in the lower right. What you see here is that the PDB structure and the neural net potential generate a conformation where there's a hydrogen bond between that emmet functionality and the heterocycle, the 5-6 system. And while the force field thinks that that's a high energy conformation. And I think what's going on here is that maybe the force field has just minor problems in angular terms. And because the system is fairly constrained in the geometry, if it were to make that interaction, it would bring the hydrogen too close to the nitrogen, and it would result in a collision. And so the only option the force field has is to flip it out of plane. So in this case, I would say the neural net potential gives us a pretty good advantage over the force field. Now, let's look at the other example where the neural net potential thinks the compounds are strained while the force field agrees with the PDB structure. And in two of the examples up here, you see we have sugar rings, highly polar systems, and that's a warning in using this neural net potential for things like strain energy calculation, in that it's trained on gas phase quantum mechanical energies. And so solvation is not taken into account. So polar interaction will be more important than in solvent in reality. The last case is actually, again, a flip of an emmet group conjugated to a 5-membered ring. I've looked at the electron density with a crystallographer at Genentech, and he agrees that the electron density could fit actually both of these conformations. I would argue that maybe the conformations favored by the neural net potential makes more sense because of the dipole interaction between the carbonyl and the NH bond over there, but that's a bit hand-wavy. In any case, this might be an example where we discovered something that warrants some more looking at in the PDB. I'm going the wrong way. Okay, what's stopping neural net potential of conquering the world and taking over where force fields have never gone before? I think there are still a little bit of couple of problems we have to work on. The current neural net potentials have no data for ionic compounds, so we need additional training data, and that's a lot of training data that needs to be done there because there's a lot of possibilities we're adding. We don't have solvations, as I mentioned before, so either we do explicit solvent or we have to figure something out. I know there are people training on QM data with solvation models. These neural net potentials are trained on data sets that have a limited number of atoms, and the most important ones are present for small molecules. But if you think about biological systems, then you also start thinking about counter ions and other atoms. So again, it adds a lot of requirements for training data. And then I want to point out what I said before. Performance of this neural net potential is amazing compared to quantum mechanics, but it's still a couple of factors lower than a force field. We've done a little bit of work to address at least one of these problems. And to introduce that, I'm showing here the workflow that is used to compute the energies in the ANI force field. You're essentially starting from atomic coordinates. For each atom, you compute the radial term, which takes into account all the neighbors around one atom within a given sphere. And you compute angular terms, which includes the atom and two of its neighbors and all combination therefrom. And you do that for each atom type and each pair of atom types in the radial term separately. You compute one descriptor for that atom, and then you feed them into neural networks that compute atomic contribution to the total energy, which you sum up and you get the total energy. And then you optimize networks to minimize the difference to the quantum mechanical calculation. One problem is that because you have this cubic angular terms and they are atom type dependent, if you double the number of atom types, you're actually going to have, so you're going to have 16 times the size of this description, and it'll make the calculation more complicated. So what we've started doing is, instead of using specific atom types, we encode the atom type in the description, and we encode it by one hot encoding the position of the atom in the periodic system, like I'm showing here. And so a hydrogen would be encoded by this 1001, and the chlorine would be encoded by 001, and then a 1 at the end. And then to encode one distance between two atoms, we have the two embeddings of the two different atoms and the distance between the two atoms. And the same thing for an angular term. We have 3 embeddings and 3 distances. That essentially describes a triangle. And if we do that, then we can, instead of calculating specific descriptors with these Bila-Parinello functions that are used in the ANI network, we're actually using a neural network to calculate the descriptors for the atom and its neighborhood relationship. But because we have the atom described as embedding instead of differentiating specific atom types, we can actually get one descriptor out that has the encoding for the atom types already in it. And we are avoiding this cubic problems with the number of atom types. If we do that, we have at the end a single network that calculates the atomic contribution for each atom, and we sum up just as in ANI. We get away with a lot less parameters. We get about the same or maybe slightly better performance than ANI. However, again, we've made the whole thing slower. At the moment, the code is about 6.5 times slower than our ANI code. And so we are still thinking about what to do there. So all the results I showed you beforehand were done with the conventional network. And with that, I have, again, the acknowledgement slide. Thank you for listening. Thanks for staying so late. And I'm happy to take questions if there are any.
Thanks, Alberto. All people online, please type your questions into the chat. I'm watching them, and we have a first question in the room. Just a second.
Hello. At the moment, you're using this only to minimize ligands, right? Do you see a potential to use these neural network energy calculations to potentially do molecular dynamic simulations in the future?
So there are people who have been doing it. Again, molecular dynamics in the terms of small molecules and proteins. Then you start talking about charges, and you start talking about counter ions, and we don't have the data yet. So there are people who are doing it. There's also John Cordera, who's using it to post-process and decalculations and improve the results by recomputing the energies of the small molecule with the neural net at snapshots in the MD calculation to get better free energy calculations. So yes, that would be the goal, but I think we are still a while away from that.
Thanks.
A better question for me. You had this long list on what still needs to be done for these approaches to really conquer the world. What's, in your experience, what about sort of like applicability domain and sort of like chemical space coverage? Can you comment on that? I mean, how many cases do you have where you have a chemistry that's too novel and not captured by the training set?
Okay, a couple of things. So the data I've showed you, the correlation that looks so good, that was applying a model that was trained on compounds with up to 8 atoms, I think, to compounds that have had, I think, 10 to 25 atoms. And so already there you're seeing you're quite outside of the usual space of the training set. I've applied it to a diverse selection of Genentech compounds. And I think I had about 12,000 or so, and I ended up, so the way they're trained, the active learning algorithm that was developed in Adrian's lab, they're using an ensemble of models to look for uncertainty, and then they explicitly recalculated compounds that have a high uncertainty in those terms. And I did kind of the same. I looked for compounds where the model was uncertain, and I recalculated about 12,000 of those, and of those, I think 120 had a fairly big deviation. So,
okay, and among the ones that were sort of like where the model was certain, did you also check some of these?
I didn't.
Okay. Yeah. But I mean, that ratio that you just mentioned is pretty good, right? It's pretty few molecules. There's a question in the chat from, oh, I'm getting old. Nice talk. Can I ask how you deal with ring conformations in your strain calculations?
So right now, it just uses whatever omega does. So if you wanted to use it, for example, on macrocycles, you would probably have to use the macrocycle version of omega and convince yourself that it does a good job. However, you don't have to find the absolute global minimum to be able to use it, right? As long as you have a low energy conformation, if you have a high strain, you know you're ways away from where you want to be. If you have a low strain, you cannot be certain that you are where you need to be. But at least in the other cases, you know that you have to do some more work.
Thanks. Another question from David Machals. Great talk. Strains matter a lot. Could you talk a bit more about the theory of the extra challenge of ionic compounds? More long-range interactions to consider?
Okay, I think that has 2 parts, right? One is the challenge in terms of strain energy, and that would be independent of the force or energy method used. And obviously you have strong interactions in charged molecules that can be shielded in solvent and can have ideal counterparts in the protein so that they counteract the energy difference to the global minimum. And so you have to allow for maybe larger ranges of strain in charged molecules. So that's one part of it. And the other part of it is the challenge in the neural networks. And that's still an open question.
Thank you. Any further questions online or in the room? Doesn't seem to be the case. So then, thank Alberto again for a great talk. And I'll try to get back here into the stop sharing. Exactly. So please online, stay online for socializing. So Andreas will put you into virtual breakout rooms. And well, I guess we say goodbye to the online world. And I hope to see you back for the next edition. Reach out to us if you would like to be presenting in this series, spread the voice about this series, and see you back in the next year.